Hi, I'm Zhijie. I'm so excited to be here to introduce my dissertation works, and thanks for your coming and listen to my presentation. And today my topic is plant-soil interaction dominate soil microbial respiration and the soil organic carbon sequestration in subtropical moist evergreen broadleaf forest in China. I will start my presentation in the following 5 contents. First, I will use a brief words to introduce my background in my dissertation. Second, I will introduce my research study areas. And third, I will give you 3 major objectives of this dissertation. Then I will show the major results in this dissertation. Finally, I will give you some conclusions of my works. So, as we know, the atmospheric CO2 concentration grows faster since industrial revolution. From this figure, we can see that the increased rate of CO2 concentration over past 50 years, as the red color in the figures, is much faster than other times. And the forest has been considering the most important ecosystem to remove the CO2 from the atmosphere through photosynthesis and retain this carbon in the tree biomass and the soil. Tropical forest is the most important in the global forest. It accounts for over 2/3 of plant biomass and 1/3 of soil carbon storage. And most important, it serviced 49% of forest carbon sink. So due to this huge forest carbon storage and the carbon sink, so even a small change of carbon dynamics in tropical forest can induce a significant change of global carbon cycling. However, in models, there still have some high disagreement to consider the carbon sinks in tropical forest. In models, the prediction of NEP variability is nearly 3 times greater than for any others. And most of this uncertainty is due to we let our understanding the below ground carbon dynamics. For example, we still let our understanding on what's the substrate carbon for soil microbes and what are the drivers of soil microbial respiration. In models, the soil microbial respiration always is based on the litter and the SOC pools and their response to temperature by the one-hole functions. In models, the litter and the SOC will divide into several carbon pools based on their carbon quality and the turnover rate. And each of these pools has its own temperature response. However, in the field, there are still other carbon sources for the soil microbes. Besides the litter and the SOC provide the energy to the soil microbes, the transport of photosynthesis from the canopy to the soil supply a huge amount of labeled carbohydrates to feed soil microbes, but most of models neglect these carbon sources. Another question, however, is that we know organic carbon decomposition is based on the soil temperature, but the photosynthesis has driven by other factors, but we're still unclear about this component. We know that a study in boreal forest found that when tree cutting stopped the transport of current photosynthate to the below ground, the soil microbial biomass and the dissolved organic carbon decreased by 41% and 45%. And also the mycorrhizal fungi biomass decreased over 52% after tree cutting. Here are two figures about the carbon storage in biomass and the soil, and the soil microbial respiration in the world forest. The tropic forest has the highest biomass in the world, but it has low soil SOC content than the temperate and the boreal forest. But tropic forest has the high soil microbial respiration. Well, this can indicating a strong link between the plant and the soil microbes in tropic forest. A comparison study from the boreal and temperate and the tropic forest found the tropical forest has much higher soil respiration in boreal and the temperate forest. And based on the author also used the 14 carbon abiotic methods found CO2 from the forest soil was much younger than the CO2 in the boreal and temperate. And the results show that over 80% of carbon in tropic forest CO2 is from the new carbon input, which is indeed tropic forest. Soil respiration in tropic forest is much more dependent on recent carbon. So another uncertainty in carbon sequestration in tropic is from the SOC change after forest conversion. As we know that for the SOC change after the forest conversion is the second largest carbon sources to the atmosphere. And most of the forest conversion is happened in tropic and subtropics. And here is the meta-analysis from the tropic forest. First, we can see that the SOC change after forest conversion is highly variability in the data. And second, we found the direction and the magnitude of change in SOC status after the forest conversion is very wide in tropic ecosystem. And then we can find that there are still lots of study sites in about the forest conversion to tree plantations. So, but in contrast with forest conversions in other countries in tropics, the transfer of the conversion of natural forest to tree plantation in China is most used type in China. As we know that China is has the largest tree plantation area in the world, and they still have the highest growing fast area in the world. The tree plantation in China account for 26.2% of the world's plantation area, and this plantation accounts for 36% of Chinese forest. And most of the plantation is Yang plantation. And the subtropical moist evergreen broadleaf forest in China is a distinctive forest type in the world. The landscape and the climate is differ with other sites in the same latitude in the world, because of the prevalence of East Asian monsoon and the uplift of Tibetan plateau. So, and also the plenty of rainfall and suitable temperature and the long growing season induced this region has the high plant productivity and the higher NEP than forests in other regions. And the tree plantation in this region is also important in China. It has the tree plantation here accounted for 63% of Chinese plantation area. And also the faster growing rate of tree plantation account for 65% of carbon sink in China. But most of this tree plantation is come from the formal natural forest. So here are 3 objectives of my presentation. First, I will try to determine the main drivers of soil microbial respiration and its carbon sources in SMEB forest in eastern China. Second, to quantify the main pathways of SOC loss during the establishment of tree plantation. Last, to test the response of bulk and the rhizosphere soil SOC to changing above and below ground carbon inputs by differing age and NPV. And my research site is located in the southeast of China, in Fujian province. The forest here is the middle subtropical broadleaf forest. For the first objective, I will start from the following 3 contents. So first, some studies have found that soil respiration is controlled by the plant productivity on annual time scale. Here is a study in tropical forest in Hawaii. The author used the several forests from the high elevation to the low elevation. This different elevation in the forest have different gross primary production. So we found that higher GPP in the low elevation has the high CO2 flux. But they study also found that the SOC didn't change among these forests. And most of the CO2 is come from the TBCF and the LIT4. So, and also in the dual scales, study also found the photosynthate is controlled the soil respiration. Here is a study in California. The author measured the soil respiration and the photosynthesis in June, July, and September. And also the results found that soil respiration is highly dependent on the photosynthesis. And the results suggest that photosynthesis provides the valuable carbon source for soil respiration. So although many of these studies have found photosynthesis is affected soil respiration, but there are still less of study to find how photosynthesis affects the soil microbial respiration. So the challenge is because we still have less of effective methods to separate the soil microbial respiration from the soil respiration. Like the traditional root trenching methods is used method to separate the microbial respiration from the soil respiration. But the material in these methods always use like the PVC bank and the thick cloth. But so this material can also exclude the input of recent photosynthate into soil, and this fresh carbon. The stop of this fresh carbon can also change the soil microbial community and the composition. Another study also used the isotopic tracing methods, but these methods is difficult to separate the RM from the soil respiration and quantify the RM rate. So because of these methods, so always like researchers just focused on assume that temperature is the major driver of RM. The third one is like we still have like our effective methods to understand the links between the plant and the microbial process. Like traditional, we measure the soil respiration like one week, 2 weeks, or 1 month per time. But we know the photosynthesis or soil respiration is highly dynamic. It's like our respiration is changed every time. So these methods it's harder to get the high links between the plant and the microbial process. So in my study, I also used the root trenching methods to separate the soil microbial respiration from the soil respiration. But here I used the very small size nylon sheet as the material to separate the fine root ingots. This small size nylon sheet can enable the water movement freely inside and outside the trenching plot. And the second, in order to separate the SOC decomposition from the soil microbial respiration, I excavate some soil cores in the field and incubate these soil cores in the open canopy without any fresh carbon input. And all this, I measured the soil microbial respiration rate by the automated measures by every 30 minutes. So these two treatments can help me to quantify the contribution of recent photosynthesis to RM. And here are 3 steps for me to quantify the rate. First, I determine the relationship between the RM and temperature in the open area to get 2 important parameters, A and B. The second step, I use the temperature in the forest, and then we calculate the RM from the SOC in the forest. And the third step, I used the difference between the RM in the forest and the RM from the SOC in the forest to quantify the RM use of the recent photosynthate as the carbon substrate carbon. Next, the data analysis, I used the wavelength transform method to transform this original data to waves different periods. These different periods of wavelength indicate different turnover rate of substrate carbon, and these different substrate have its own environmental drivers. So in my results, first Here speak about my start. I mentioned in this study. The black curve is the RM in the forest, and the gray curve is the RM in the open. We can see that the RM in the forest is much higher than RM in the open. So also based on the equations I mentioned before so we found that the recent photosynthesis contributes 88% of RM in this forest. And the second, we can find the black curve has high dynamics in dial and seasonal change, but the gray curve also is more stable in the year. But we can see from the temperature dynamic, we can see first the temperature in the open is much higher than the soil temperature in the forest. And second, soil temperature in the open is much dynamic than the soil temperature in the forest. And next, I used the global wavelet power spectrum to looking for the carbon sources to the RM. So first, I found the 24-hour period was the only prominent frequency for both RM in the forest and the RM from the current photosynthate. And second, I found that the no signal variation at the dial period for the soil cost in the open, which indicate that soil RM in the forest and in the open has different carbon sources. Then I used the local wavelet power spectrum to find how when this 24-hour happened in the year. The red color in the the red color with the contour line indicate the high power with statistical significance. We can see that most of this 24-hour is happened from the April to the July in the growing season, and this 24-hour period also corresponds the high RM in the year. Then I used the wavelet coherence to find the cause effect between the RM and the biological factors. So also in these two figures, I run the relationship between the RM and the PAR and the soil temperature. The red color with the black contour means the high correlation with statistic significance. And also the second one also is important is that we can find many errors in the red color inside the red color. At least the arrow direction is important for us to see how this like when the arrow goes up means that environmental factors lead the RM, which means that the environment factor affected is the driver of the RM. So first, I found the RM was tightly correlated with the PAR and the soil temperature both at the 24 period. But based on the arrow direction, I found the RM is lagged the PAR by 2 hours in the growing season and 60.5 hours in the drought season. And last, I found the RM leading the soil temperature by 1.8 hours in most of the time. So based on this analysis, I found that the PAR is the major driver of the RM in this forest. So I get a summary of this, the first chapter study. I found first, the current photosynthate are the primary carbon sources for microbial respiration in the SMEB forest and contributing 88% of carbon source for microbial respiration. And second, the dynamics of soil microbial respiration was strongly controlled by power, but not soil temperature in models. And for the second object, to quantify the main pathways of SOC losses during the establishment of tree plantation. First, I would like to introduce the major tree plantation species in subtropical forest. It's the Chinese fir plantation. It's widely distributed in the southern of China, and this tree species is the largest monospecific plantation type in China, and its area is 1.12 million hectares in China and accounted for 30% of Chinese plantation area. And also this forest is growing very fast. Like it always can grow, its height can grow 1 meter per year. And in order to, when we plan to establish the tree plantation, first there are some steps for us to do. First, we harvest the natural forest and remove all the trunk and left all the harvest residue on the surface soil, and dried it on the ground under the sun for several months. Then we start burning this harvest before seedling. And also we had a lot of intense disturbance on the soil, like dig hole for the seedling and also weeding. And this intense or human activity can induce different pathway of SOC losses. Like the sludge burning can induce lots of carbon losses from the carbon volatilization. And also, because here we have plenty of rainfall, and also the landscape here is very steep. The steep slope here is the average of slope here is 27 degrees. So this high amount of rainfall and the steep slope can use lots of SOC loss from the soil and the water erosion. Finally, the last one is because the intense of human activity can disturb the soil structure, so this can increase the SOC decomposition. So although we found that this human disturbance is important to quantify the SOC losses, but there are still few studies have quantified these SOC losses in the field. So in this study, my group has established these sites in 2012. In preparing for plantations, we harvest a natural second forest in Fujian province, and based on like clear cut and slash burning. Then we planted the seedling in 2012 after the slash burning. And also in this study, I chose the major Chinese fir plantation and the natural secondary forest as the control plots. Here is some pictures about the development of the tree plantation since 2012. The tree is growing very fast. We can see that after 3 years, all the canopy is recovered. So in my results, first here are two figures about SOC concentration and SOC stock changed after the sledge burning. First, I found the SOC concentration decreased by 15% immediately following sledge burning. This can calculate as representing a loss of 3 ton carbon by the sledge burning. And also, I found 2.6 ton carbon losses from the soil erosion in the first year after the forest conversion. And third, I also measured the soil microbial respiration between the NRS and the YCF. I found most of the young transfer plantation has high SOC decomposition in the first half year, and this will induce the SOC losses of 2 ton carbon from the increase of SOC decomposition. So in the summary, I found in the first year of the forest conversion, there are less 28% of SOC was lost from the 3 pathways, and the sledge burning contributed the most of SOC loss. Then I also measured the soil microbial respiration in the second year by the high frequent measurements by every 30 minutes. I explained it in the last chapter. Between these two, in these two forests, the NRS and the YCF, I found first they are different. They have carbon sources from the 24 hours, but the young Chinese plantation also has a power at the long period, which indicate the YCF also accelerate the SOC decomposition in the second year. The next, I also collected the soil samples from from 3 times in the different years in the young Chinese plantation and major plantation and the NRS. And my results found that first, the Chinese fir plantation is hardly recovered to the level of the agent natural secondary forest. And also the SOC didn't change it, although the Chinese fir derived from the young to the major forest. So here is a summary of this chapter. First, I found the conversion of natural forest to tree plantation induced SOC losses by 28% in the first year after conversion. And most of the SOC losses were the result of sledge burning. Third, the conversion of natural forest to tree plantation also accelerated the SOC decomposition. And SOC didn't recover within 36 years, like the tree plantation rotation in this region. So for the third objective, to test the response of bulk and the rhizosphere SOC to changing above and below ground carbon inputs. I will study like first, as we introduced before, like soil microbial activity is highly dependent on recent carbon inputs like current photo or literal. But most models use temperature to predict the SOC change. So we need to get more information about how these bio inputs to affect the SOC changes. And from some some studies have found that SOC sequestration is also influenced by the plant productivity. Like the results found the high MPP will enhance the amount of carbon inputs into the soil. This input can result in shifts in microbial activity and the community composition. But this increase of carbon inputs can also accelerate the SO decomposition to meet the enhanced plant demand for nutrition. This can also change the SOC sequestration and the composition. And the study also found that SOC changes limited SOC changes by the increase of MPP in the actual mycorrhizal fungi species in temperate forest. This is because the increase of carbon inputs by the high MPP also stimulate the nitrogen-related enzymes activity and accelerate SO decomposition. So leads to get a new balance that induce the SOC didn't changes although the increase of MPP. But less of information about the response of SOC sequestration to the MPP in tropical and subtropical forest compared to the temperate forest. And also here are some difference among species and the forest types. Like first, in the temperate forest, it's always think the plant growth is limited by the nitrogen availability. But in tropical and subtropical, this forest is limited by the photosynthetic availability. And also for enzyme species, like in the EFM species, can form enzyme to decompose SOM for nutrients. But the AMF species has lower subtropic capability to decompose SOM. And another important is because we also know that the bulk soil and the rhizosphere soil has different carbon substrate carbon for the different quality of substrate carbon. Like in bulk soil, always from the little in the rhizosphere soil, like they have much of carbohydrates from the root exudes. This induces the low efficiency in bulk soil and the low soil microbial abundance. So in this study, I chose 3 different development of Chinese fir plantation from the 6-year-old and 18 years old and 42 years old. These different forest ages has different MPP. Like in 18 years old has the highest MPP, and the young and the mature plantation has the similar MPP, but they have different carbon allocation. Like in mature plantation, it has higher little but low below ground carbon allocation. And then I collect soil samples from the rhizosphere and the bulk soil in these three plantation. And also I measured the soil SOC fractions in the lab. I separate the SOC to 3 fractions. First is labile carbon per 1. It also consists of the non-cellulose polysaccharides. And the labile carbon per 2 consists of cellulose. And last is the recalcitrant carbon per always from the plant-derived lignin and the microbial residues. Also, I matched the total and the available nitrogen, phosphorus, and also soil microbial biomass and carbon, and also then 6 soil enzymes is related to carbon, nitrogen, and the phosphorus. So in my results, first I found that SOC concentration differ with a standard age in rhizosphere soil, but not in bulk soil. And the middle age plantation has the lowest SOC, also nitrogen and phosphorus, and with the highest MPP in. So in soil fractions, first I found that label carbon pool 1 and the recalcitrant carbon are the primary SOC pools in all plantations. And the second, I found the young plantation had the highest label pool 1 carbon than the middle-aged and the major plantation. But the major plantation had the highest recalcitrant carbon pools in rhizosphere soil. Then I used the redundancy analysis to analyze which factors affect the SOC change and the land fractions. So first in the bulk soils, I found the total phosphorus explained 69% of the variance among the forests of different ages. But the relationship between the SOC and the environment facts across the different age stands were not apparent. And in rhizosphere soils, the total phosphorus and the microbial biomass carbon to microbial biomass nitrogen ratio explained 72.7% and 20% of the variance of SOC variation among forest ages. And the microbial biomass carbon to nitrogen ratio and the phenol oxidase were positive, correlated with the label carbon pool 1 in young stands. And the total phosphorus was correlated with the recalcitrant carbon in mature forest. Then the dynamics of SOC were correlated with the MPP in middle age standards. So in this chapter, I found the forest productivity across different growing stages, control nutrient availabilities, and the soil microbial activity in the rhizosphere, and thus regulates the quantity and the quality of rhizosphere SOC. The second, soil phosphorus and MBC to MBN ratio play the important roles in bulk and the rhizosphere SOC, which draw the label and the recalcitrant SOC in rhizosphere soil in different ways, depending on forest age. So, I'm sorry. Here's my conclusions on my study. First, I found that main drivers of soil microbial respiration in SNEB forest is recent photosynthate, which is controlled by the PAR. And models using the response of SOC to temperature to predict RM are not effective in tropic and subtropic forests. And second, we all know that the faster growing tree plantation is the most important carbon sink to fighting the global change. But my result found these tree plantations are hugely problematic for carbon sequestration due to the dramatic reduction in SOC by stage burning during the establishment, which offsets the biomass carbon accumulation in faster growing tree plantations. So, and also I found the SOC didn't accumulate to pre-conversion levels, showing the need for a new approach to plantation management. A third, I found that the interaction between the plant, microbe, and soils dominate the variation in rhizosphere SOC content and composition across different growing stage of plantation. So it was stating the influence of the root microbial interaction on SOC sequestration can improve our understanding of the long-term SOC pro dynamics and reduce the uncertainty of SOC prediction in climate models. So finally, I'd like to thank everyone who has helped me in this project, and also thanks again for you coming up for here to listen my presentation. Thank you.